Very good evening, children. Very good evening to all. Can you all hear me? Can you all hear me, children? Okay, children. Okay, thank you. We'll start our today's topic. Today's topic is reproduction in flowering plants. Reproduction in flowering plants. That is the topic for today's class. So we are going to start this topic. This is a new chapter. First, let me explain about the reproductive part of a plant. The reproductive part of a plant it is the flowers. The reproductive part of the plant is flowers. The flowers are the reproductive part. They are the reproductive part. There are basically, we all know that there are 4 parts of the flower. We call it as calyx, corolla. Calyx are, and then we have androecium. This is called as sepals. And petals. Androecium are stamens. And then gynoecium are pistil. These are all the 4 important parts of a flower. And flower is the reproductive part. Among these two parts are called as non-essential part. These 2 parts are called as non-essential part. Non-essential part. And these 2 are called as essential part. So why they are called as non-essential essential part? Because these 2 parts are involved in the process of reproduction. These parts are in the process of reproduction. That's why they are known as the reproductive part. Here the androecium, it is the male reproductive part, and gynoecium is the female reproductive part. Female reproductive part. So androecium and gynoecium. Androecium and gynoecium. So now let us see about the topics in detail. So first, I've given a general information about the reproduction in flowers. Now let us see about that. So these are the 4 important parts of the flower. We have calyx, corolla, androecium and gynoecium. Calyx, corolla, androecium and gynoecium. So you can see it here. The androecium and gynoecium are essential part, and non-essential parts are calyx and corolla. And you can know the functions of the calyx. Calyx are sepals are the outermost whorl of the flower, outermost layer of the flower, and it is green in color. So it also does the process called photosynthesis. And in the young stage, the whole calyx or sepals it covers the other whorls of the flower. In the bud condition, the calyx protects the other whorls of the flower. That is about that. Next is called corolla. The colored part of the plant, the colored part of the flower, we call it as corolla or petals. And these are mainly to attract insects for pollination. Insects for pollination. Now, I'm going to explain to you about the androecium and microsporogenesis. We all know that androecium, it is the male reproductive part. The male reproductive part is androecium, and the term called microsporogenesis, that means formation of the microspores or the pollen grains from the microsporangium, that is called as microspores. So let me explain about that. What is androecium? Androecium, it has two parts. This is the male reproductive part. It has the filament and anther. The filament and anther. This is filament. And this, the kidney shaped part, we call it as anther. We call it as anther. So anther and filament. Anther and filament. So this anther is very important part, because this anther, it contains microsporangium. Microsporangium. What is this microsporangium? This is the mother cell. The microsporangium is the mother cell that produces the microspores. Produces the microspores. So these microspores, it differentiate and becomes the pollen grains. Pollen grains. The pollen grains contains the male gamete. Contains the male gamete. So now we know what is male gamete. Male gamete, it is present in the pollen grains. Pollen grains are nothing but the microspores. The microspores are produced by the microsporangium, which is present inside the anther. So now I'm going to explain about that. So these two things, the anther and filament, all together we call it as stamens. We call it as stamens. Anther and filament all together we call it as stamens. Now I'm going to explain the process to you in detail. So now we can move on and learn about the androecium and microsporogenesis. Children, can you all hear me? Am I audible to all? Am I audible to all? Okay, children. Okay. So now we'll move on to androecium and microsporogenesis. I told you about androecium. This is the male reproductive part. It consists of group of stamens. Group of stamens. So each stamen, it has a stalk. This is the stalk. The stalk is called as filament, and the tip, we call it as anther. This is called as anther. The anther, if you take the cross section of the anther, it will be a tetrad structure like this. It will be like this. And inside the anther, it has pollen sac. This is called as pollen sac. So that pollen sac, it has the microsporangium. The microsporangium. What is microsporangium? The pollen mother cell that produces microspores. The pollen mother cells that produces microspores are called as microsporangium. Now, I'm going to explain to you about the development of anther, how anther is formed. This is the typical structure of the anther, how it is formed. I'm going to explain to you about that. And till I'm going to explain to you about the development of male gamete. And then we are going to talk about gynoecium or megasporogenesis. I'll go a little bit slow so that everybody can be able to understand this process. First, we're going to talk about the development of anther. Development of anther. The anthers are mainly formed from homogeneous mass of cells. These are all the homogeneous mass of cells that are surrounded by epidermis. In a young anther, it has homogeneous cells. The homogeneous mass of cells are surrounded by anther. And during the development of anther, the few rows of the cells, the hypodermal cells. What is hypodermal cells? The cells which are lower. This is called hypodermal cells. The anther, the homogeneous mass of cells are surrounded by epidermis. During the development, the anther, it forms a four-lobe structure. That's what I told you about four-lobe structure. It will be like this. So the four-lobe structure formation only I'm going to explain to you now. How that process takes place. Here, each lobe, a few rows of hypodermal cells, they becomes enlarged. They develop nuclei and becomes archisporial cells. So now we can see it here, the homogeneous mass of cells, it forms archisporial cells. You can see that they form four archisporial cells. You got it? The homogeneous mass of cells, the few layers of hypodermal cells, they become enlarged, they form a conspicuous nucleus, a well-developed nucleus, and then they become archisporium or archisporial cells. These archisporial cells, it starts dividing. There are two different types of divisions here. One is called periclinial division and anticlinial divisions. So they undergo the division process. Either it can be periclinial division or anticlinial divisions. Here what will happen? The archisporial cells that are formed here, it divides periclinial like this, periclinial, to form primary parietal cells. Now what will happen here? The archisporial cells, it will become primary parietal cells. And there are two types of divisions it can take place. One is called periclinial divisions. Here, this division, it is parallel to the tissues or organ surface, like this parallelly. This one is right angle to that. Right angle to the tissue or organ surface. So now what will happen here? The homogeneous cells, it become archisporial cells. The archisporial cells divides and it forms primary sporogenous cells. Now, the primary parietal cells, the primary cells which is formed here, it undergo a lot of divisions. It undergo a series of divisions. It can be both periclinial or anticlinial divisions, and they form 2 to 5 layers of anther walls. So this structure, it undergoes divisions and finally it forms structure like this. So this structure, it is formed by series of divisions. The series of divisions, both the divisions can be periclinial divisions and anticlinial divisions. So the homogeneous cells, it become archisporial cells. Archisporial cells divides and become primary parietal cells. And they also form primary sporogenous cells towards the inside. So outside, it forms primary parietal cell. And inside, it forms primary sporogenous cells. So two cells are formed here by the archisporial cells. The homogeneous mass of cells, it becomes archisporial cells. The archisporial cells, it divides and forms two types of cells. One is called primary parietal cells. The other one is primary sporogenous cells. So this primary parietal cells, it undergo a lot of divisions and form the layers. So this is the typical structure of the sporogenous cells or the microsporangium. We can see that it has endothecium, the outer layer, and then middle layers. These are all the middle layers, and tapetum. This is tapetum. Whereas the center, this is the primary sporogenous cells. So these epidermal or endothecium, middle layer, and tapetum, they are all formed by primary parietal cells. They are formed by primary parietal cells. Whereas the primary sporogenous cells, which is here, this is the primary sporogenous cells. You got it, children? Till this, are you all clear? Till this, children, are you all clear? You want me to explain one more time? இது வரைக்கும் எல்லாருக்கும் clarity இருக்காமா? What about others? Ajravan, Vashini, Madev? Yes. Okay, very good, Vashini. I'll explain one more time. Okay. So first, what will happen here? I'm going to explain one by one. Okay. Can you all see it here? Now, first, what will happen here? The homogeneous cells, the homogeneous mass of cells. The homogeneous mass of cells, it will become, it undergo the modification and it forms archisporial cells. Archisporium, or it forms archisporial cells. So now what will happen? The mass of cells, it will become a tetrad structure like this. They will form a structure like this. And now we have the archisporium. 4 archisporiums are developed. 4 archisporiums are developed. So these archisporial cells, it undergo divisions. It undergo divisions. It undergo divisions. The divisions can be divisions can be periclinal division. Either it can be periclinal division. The division can be like this. Periclinal division. Periclinal division, or it can be anticlinal divisions. Anticlinal divisions. So these archisporial cells, archisporium, it undergo divisions. It undergoes several divisions. And these archisporial cells, it undergo divisions. And these archisporial cells, it forms 2 structures. Outer, it forms certain structure, whereas inner, they form other structures. In the outer, they form primary parietal cells. Primary parietal cell. Whereas inside, it forms primary sporogenous cells. So now we can see here the archisporium, it will become 2 different types of cells. One is called the divisions are primary they form either primary parietal cells, the outer and inner, they form primary sporogenous cells. They form primary sporogenous cells. Now what will happen? The primary parietal cells, the primary parietal cells, the primary parietal, the outer layer, it forms three structures. It forms three structures. One is the endothelium or epidermis. We call it as epidermis or endothelium. And middle layers and the innermost layer, that's called tapetum. Tapetum. So three layers are formed. Whereas the primary sporogenous cells, the primary sporogenous cells, it forms the sporogenous tissue. It forms the sporogenous cells. It forms the sporogenous cells. Now, what is going to happen to this now? This is about the structures that are formed there. These are all the four structures. The structures that are formed. So you can see it here. These are the typical structures that are formed. This is actually called as the microsporangium. The microsporangium are pollen mother cells. The pollen mother cells. Now, what is going to happen here? You can see that. Now, I'm going to explain to you about the microsporogenesis process. The microsporogenesis process. You can see that is a typical structure of the anther, where you can see the sporogenous cells. The sporogenous cells are surrounded by tapetum, middle layers, endothelium, and outer they have epidermis. For all the four structures, it will be like this: epidermis, endothelium, middle layers, tapetum, and inside sporogenous cells it will be there. Okay. So now I'm going to explain to you about the microsporogenesis process. Microsporogenesis process. In the microsporogenesis process, what will happen here? What is microsporogenesis? The formation of microspores. The formation of microspores are known as microsporogenesis. What is this microspore? I have told you already, it is nothing but the pollen grains. This is the microsporocyte. The microsporocyte, which is present inside the microsporangium. Here, the primary sporogenous cell. This is the primary sporogenous cells, right? Same the primary sporogenous cells, it undergo many mitotic divisions. It undergo mitotic divisions and they form microspore mother cells. Microspore mother cells. So here, the primary sporogenous cells, that is sporogenous cells, it undergo mitotic divisions and it forms microspores, which is called as the primary microspores. They form the primary primary microspore is nothing but the microspore mother cells. They form the sporogenous cells, it undergo mitotic divisions and it forms the microspore mother cells. These microspore mother cells, the microspore mother cells, Now it undergo the meiosis process. First, you can see that the sporogenous cells, it undergo mitotic process and it forms microspore. These microspores are diploid. Now these microspores, it undergo the process called meiosis. So meiosis process, the microspores, it produces 4 haploid microspores. It forms 4 haploid microspores. So the microspore mother cells, it undergo the divisions, meiotic divisions. The meiotic division, we know that it has 2 phases. That is meiosis 1 and meiosis 2. The meiosis, the diploid cell, it undergo divisions and it forms 4 haploid cells. It forms 4 haploid cells. So these are the 4 haploid cells. Those 4 haploid cells which are formed here, we call it as haploid microspores, or we call it as microspore tetrads. They are also called as microspore tetrads. They're known as microspore tetrad. And these microspore tetrads, there are actually, there'll be like 4 cells, it will be there. So I'll show the diagram for you to understand. How it will divide, you can see it here. This is the microspore. First it divides 2 and it becomes 4. And these are all the microspore. We call it as microspore tetrad. 4 spores are formed. All of them are haploid. So these microspores, which are here 4, it gets separated and they develop into pollen grains. They develop into pollen grains. Again, I'll repeat, the microspore, it undergoes meiosis and forms 2 cells first, and then become 4 haploid cells. These 4 haploid cells are called as microspores. They are called microspore tetrad. These microspore tetrads get separated and they develop into pollen grains. Okay, in some plants, the microspores are not separated. They are like 4. They are joined together. That is called pollinium. Pollinium means the microspores are attached, remain attached. Here, the formation of pollen grain, the 4 microspores, it gets separated and become pollen grains. Whereas in the case of pollinium, the cells, it remains held together. This is the transverse section of matured anther. Okay. Yes, Shruti. Sir, why does the sporogenous cells undergo mitotic divisions and other don't undergo it? Okay. The sporogenous cells are the one which is supposed to form the anthers, the mother cells, which are supposed to form the mother cells, the microspores. Whereas the other cells, the primary cells they're going to form only the outer layers. The outer layers. It also divides mitotically, but only sporogenous cells, it undergo mitosis as well as meiosis. Mitosis as well as meiosis. Okay, now this is the transverse section of matured anther. You can see that sporogenous cells, they are surrounded immediately by tapetum. This is the sporogenous cells, they are surrounded by tapetum, and then middle layers, endothecium, outer is epidermis. You can see the whole structure, tetrad structure. So this structure of anther, we call it as tetrasporogenate, because there are 4 sporogenous cells are there. 1, 2, 3, 4. That's why it's called tetrasporogenate. And you can see that layers. The outer layer is the epidermis, and then endothecium, middle layers, and tapetum. So what is the outermost layer in epidermis function? It is a single layer and it is protective. Epi means outer, dermis means skin. It is only protection. Whereas the endothecium, the endothelium, it is next layer. Can you see it here? The layer below the epidermis, that is called endothelium. And this endothelium, it forms a fibrous thickening, because endotheliums are made up of cellulose, pectin, and lignin. Three substances, cellulose, pectin, and lignin. So mainly the endothelium, it helps in dehiscence of anther. What is dehiscence of anther? During the development of pollen grains, the whole pollen grains development takes place within the anther. And the anther, once the pollen grains are matured, the anther, it will split open. It will burst and releases the pollen grains. You got it? The anther will just remain like this. Once the pollen grains are formed inside, once the pollen grains are matured, it ruptures. That is called dehiscence, the breaking. It dehisces and releases the pollen grains also. And next is the middle layers. So you can see the middle layers here. Next to endothelium, we have middle layers. They are the functions of the middle layers. Mainly, it provides nourishment to growing microspore mothers. Partially, these middle layers, they live for short time. They are ephemerals. They provide nourishment to the microspore mother cells. Whereas mainly the tapetum, which is the layer that surrounds the sporogenous cells. The tapetum, it is a specialized layer of nutritive cells. A special layer of nutritive cell that surrounds the anther, that surrounds the microspore mother cell. And they provide nutrition, and also they play an important role in development of pollen grains, that is from the microspore to pollen grains. Among this, the tapetum is a very important layer because it provides nutrition to the microspore mother cells, and also they play an important role in development of pollen grains. And then the pollen coat. What is pollen coat? The pollen coat are the outer layer of the pollen grains. The outer layer of the pollen grains, it has two things. One is called exine and intine. The pollen grains, the functions of the tapetum. The functions of the tapetum, I'll just list out here. The tapetum, it has certain functions. One is it provides nutrition to the microspores. It provides nutrition to the microspore mother cells. Second function, helps in development of pollen grains. Helps in development of pollen grains. And third function, the tapetum, it helps in development of pollen grains, in that the overall development of pollen grains, mainly the pollen coat. The pollen coat. What is pollen coat? If you see the pollen grain, it has two outer layers. The outer layer, we call it as exine, and the inner layer, it is called intine. These are all the pollen coat. So the formation of these pollen coats, the tapetum play an important role. The tapetum play an important role in that. Now I'm going to talk to you about, okay, the I want to explain the other functions also. I told you about the epidermis. The epidermis. The epidermis, it is the outermost layer, mainly it is protection. Whereas the other layer, we call it as endothelium. The endothelium, it tells in dehiscence of anther, dehiscence of anther, splitting up of anther. Here, you all know that there is a filament and anther. Stamens contain filaments and anther. Within the anther only what will happen? The pollen grains are formed. The pollen grains are formed. That process we all know that it is called the microsporogenesis. The pollen grains are formed. Once the pollen grains are formed, these pollen grains, once is formed inside the anther, once the pollen grains are matured, the anther splits. Anther splits. The anther breaks and release the pollens. Release the pollen grains. Anther splits and release the pollens. So that process of releasing the pollen, that is called dehiscence of anther. That is done by endothelium. Endothelium. Similarly, the other layer, they are all called middle layers. The middle layers are short-lived. They are all short-lived. And these also provide nourishment, nutrients to the microspore mother cells. Microspore mother cells. These are the functions of the 4 layers. Now, I'm going to explain about the types of tapetum. There are 2 types of tapetum are there. One is called secretory tapetum and invasive tapetum. Secretory tapetum. This secretory tapetum, it provides nourishment to the microspores, whereas the invasive tapetum, that is one more type is there, which is called invasive tapetum. This invasive tapetum, it fuses among themselves. Those tapetum, it fuses among themselves and they form tapetal periplasmodium. They form the tapetal periplasmodium, and this tapetal periplasmodium only, it surrounds the pollen mother cells and helps in development of pollen grains. I have told about the functions of the pollen grains. In that I have told they help in development of pollen grains. So these are that invasive tapetum. The invasive tapetum, it forms joined together and they form a tapetal periplasmodium. This tapetal periplasmodium, that tapetal periplasmodium only, it surrounds the surrounds the pollen mother cells or the microspore mother cells and helps in development of pollen grains. These are the 2 types of tapetum. Next, these are the microspores and pollen grains. So now we know that 4 microspores are formed. These 4 microspores, they get separated and they form pollen grain. This is a typical structure of the pollen grain, where it forms the outer layer called exine and inner layer intine, and then vegetative cell and generative cells. They'll have a large vegetative cell and a small generative cells. The exine, the outer layer, it is made up of sporopollenin, and inner layer is called intine. It is made up of cellulose and pectin. It is made up of cellulose and pectin. So this is about the structure of pollen grains, and these are all how the microspores spores are formed and how they are transformed into pollen grains. This process we call it as microsporogenesis. Okay, children. So now just draw this diagram. I'll give you 5 minutes time. Take a break for 5 minutes. Then come back after 5 minutes where I'm going to explain our development of the male gametophyte. How the male gametophytes are developed. And then I'm going to explain to you about the gynoecium or megasporogenesis. How the megaspores are formed. Okay, children. I'll explain that after a short break. Now, just draw this diagram and take a break. I'll come back within 5 minutes. Shruti, the outer layer of pollen grains is different from epidermis. The epidermis is total anther. For that, the outer layer is epidermis. For pollen grains, which is formed inside the pollen sac, inside the porogenous cells, right? For that only intine and exine is there. The epidermis, it is for the whole anther, whereas the exine and intine, it is for the pollen grains. Children, are you all there? Shall we start? Children, are you all there? Shall we start? Okay, ma. So we'll move on to the next topic. The next topic is development of male gametophyte. Development of male gametophyte. What is the development of male gametophyte? Here, from the microspore, this is the microspore. From the microspore, how it develops into male gamete or develops into pollen grain, and how they become the male gamete. You can see the series of changes: 1, 2, 3, 4, 5. These changes, we call it as development of male gametophyte. Development of male gametophyte. So let me explain about that. Now all of you take notes. The next topic is development of male gametophyte. In development of male gametophyte, what will happen here? The microspore, which is a haploid cells, this microspore, which is a haploid cell. Haploid cell. What is haploid cells? N number. This is the microspore. The microspore, which is a haploid cell, mainly the nucleus. The nucleus of the microspore or haploid cell divides. The nucleus of the microspore, it divides into two. It divides two. And the divided two nucleus, they form two types of nucleus here. One is called vegetative nucleus and generative nucleus. The nucleus of the microspore, it divides into two and they form vegetative nucleus and generative nucleus. And here there are two nucleus are formed. In that two nucleus, the vegetative cells, it contains abundant food reserve. It has abundant food reserve, so it will transform like this. It will transform into one large cell which abundant of nucleus, abundant of food substances and nucleus. That's called vegetative cell, and a small generative cell is formed. Now you can see it here. So first the nucleus divides. Now the nucleus, it transforms and it becomes two cells. So two unequal cells are formed. One, it will be large cell, that is called vegetative cell. The other one, it will be smaller cell, that is called generative cells. In this condition only, the pollen grains are degassed out. The pollen grains are released only in these conditions. The microspore, the nucleus of the microspore divides and becomes vegetative and generative nucleus. And now the cell, it divides into large cell, which is called vegetative cell, and small cell, that is called generative cell. So this is the vegetative cell, and this is the generative cells. The two cells are formed. One is called the vegetative cell, the other one is called the generative cells are there. And this is how it will be released out. Now, the second scenario, further development of the pollen. The further development of pollen grain, it occurs after pollination. After pollination. So almost all the pollen grains that are liberated from the anther, that are sent out from the anther, it will be like this. It will have the vegetative cell, the generative cell, and also they have the outer layers, that is the outer layer, and then there will be a small pore here. This is called germination pore. The outer layer is called exine, and inner layer, which is called intine, and then a large vegetative cell and a small generative cell, it will be there. Also contains the outer layer, exine and intine. So now the pollen is released out. Now what will happen here? After the further development, it takes place after pollination. After pollination. What is pollination? Transfer of pollen grain from anther to the stigma. Transfer of pollen grains from anther to the stigma. It is known as pollination. It is known as pollination. So transfer of pollen grains from anther to the stigma. Now, assume that after pollination, the pollen grains reach the stigma. So this is the stigma of the gynoecium, and this is the pollen tube, and this is the ovary. Okay, this tip is called a stigma. So the pollination process, the pollen grains reaches the stigma, and this is called ovary. Now what will happen here? The pollen grains, it reaches the stigma here. It reaches the stigma. The pollen grains, it reaches the stigma. That process is called pollination. So in this structure only, the pollen grain reaches the stigma. Once the pollen grain reaches the stigma, it undergo the process, the further developmental process. Here, once the pollen grain reaches the stigma, it does two things. One is it absorbs moisture. It absorbs moisture, and once it absorbs the moisture, we know what will happen here. It swells up. It swells up. Here, once the pollen grain reaches here, it absorbs the moisture and it swells up. it increases in size. It increases in size. And after, what will happen here? The intine. What is the intine? The innermost layer. The innermost layer. You know that there are two layers are there. The innermost layer, the intine, it grows as pollen tube. It grows as pollen tube. Where these pollen tubes are formed, think that this is the pollen grain. This is the pollen grain. The innermost layer, it forms a tube-like structure which is called the pollen tube. The innermost layer, that is intine, it forms the pollen tube. The pollen tubes are formed. So this pollen tube, it grows inside the style. Once the pollen grain reaches the stigma, it absorbs the moisture and swells up. Once it swells up, the innermost layer, intine, the innermost layer, this is the intine, right? The intine, the innermost layer, it forms the pollen tube. It forms the pollen tube. And this pollen tube, it grows through this pore. This is called the germination pore. The intine, it grows through the germination pore. Now what will happen? Inside that, now the pollen tube here, what you are seeing here, the pollen tubes are formed by the intine. Inside that, what will happen? The generative cell divides. The generative cell, it grows inside the pollen tube. The generative cells, the generative nucleus, it divides and form two male gametes. Inside the pollen tube, the generative nucleus divides and forms two male gametes. So usually two male gametes, it will enter into embryo sac. What is embryo sac? It is a structure that is formed inside the ovary that contains the female gamete. Here you can see that the generative cells, the nucleus of the generative cell, it divides. The nucleus of the generative cell, it divides and it forms the male gametes. So what is the purpose of absorbing moisture? Any germinating cells, it absorbs moisture for them to grow. Any cell, it absorbs the moisture and it develops into, it swells up. That is the growth actually. Even if you put seed inside the soil, it takes water. That's called imbibes water and it grows. For the growth, it needs a lot of moisture. And what is the difference between the gamete and the gametophyte? Gametophyte, it is a plant structure or generation of plant. To take plant, it has two generations. One is called vegetative generation or vegetative growth and gametophytic growth, or it's called sporophytic growth and gametophytic growth. The gametophyte are the plant that develop the gametes. In plants, the gametophytic structure of the plant, the gametophytic structure of the plant, the gametophyte structure of the plant, it has two structures, which is called as archegonium and antheridium. Archegonium and antheridium. The archegonium, it is the female reproductive structure, a female part, and antheridium is male. And this archegonium, it produces egg, which is the female gamete. Similarly, antheridium, it produces anthrozooids, which are the male gamete. Similarly, in higher organisms, in humans like that, we have testis and ovary. The testis produces sperms and ovary produces ovum. So this is the gametophyte. Gametophyte, it is a generation in plants. Generation. There are two types of generation is there. One is called sporophytic generation, which produces spores, and the other generation is gametophytic. In gametophytic generation, they produces gametes. So how this is actually a cycle that you can see it in the case of organisms. In the case of organisms, how it will be: the sporophytes, in sporophytic generation, it produces spores. The spores develop into gametophyte plant. This gametophyte produces gametes. These gametes, it forms the sporophytic generation. There are a lot of, in between, there are a lot of changes are there. I'm not explaining that. Sporophytic generation give rise to spores. Gametophytic generation give rise to gametes. I hope you have understood what is gametophyte and gametes. And next is the pollen grains. Actually, the whole pollen grains does not contain the gamete. As I mentioned earlier, only the germ cells. The germ cells, it forms the male gamete. So basically, any cell or any pollen grains that is growing towards the ovary, it has two male gametes. It has two male gametes. Next, I'm going to explain to you about gynoecium or megasporogenesis. Gynoecium. We know about gynoecium. That is the female reproductive part. This is the typical structure of the gynoecium, and it has the tip called stigma and the hollow tube like this. It will be there. That is called style. The hollow structure is called stylar canal, and then this part is called ovary. And inside the ovary, there are ovules are there. The ovules are attached to the ovary with the help of placenta, and you can see the locules here. Locules means divisions. So the stigma, style, ovary. Inside the ovary, ovules are there. And then placenta. Placenta, it will be there. This is a gynoecium. Now, I'm going to explain to you about the development of the female reproductive structure. This is the typical structure of the ovary. And you can see these are all ovules. The ovules are attached to the ovary with the help of placenta. Placenta. The ovary, inside the ovary, ovules are there. So this is one ovule. In that ovule, inside that, there will be embryo sac. You got it? You will have the ovary. Inside the ovary, ovules are there. Inside the ovules, embryo sac is there. Understand clearly, children? This is the ovary. Inside that ovary, ovules are there, and ovules contains embryo sac. So within the embryo sac only, the female gamete or egg cell is present. Okay, now I want to explain to you about the development of embryo sac. Here what you are seeing is not embryo sac. The embryo sac, it will be a typical structure like this. This is actually the embryo sac. This is the embryo sac structure. How this structure is formed, I am going to explain to you about that. That is called the development of female gametophyte. Development of female gametophyte. I am going to explain to you about that. So now, inside the ovary, the ovules are there. Inside the ovules, it has megaspore mother cell. Megaspore mother cell. That megaspore mother cell are surrounded by nucellus. The nucellus are surrounded by integuments. Okay, now just listen. Inside the ovule, for the development of embryo sac, what is required? Megaspore mother cell is required. Now I am going to explain that. Like how the homogeneous cells, it forms archisporium. Here what will happen? A similar process takes place here. That is called megasporogenesis. The microsporogenesis, the homogeneous mass of cells, it forms the archisporium. Archisporium. Whereas here, the nucellus. What is nucellus? The cells that surrounds the megaspore mother cells. The nucellus, this is the nucellus, becomes enlarged. A single nucellus cell, it becomes enlarged and they develop into archisporial cells or archisporium. The nucellus, in that nucellus cells, one nucellus cells, it enlarges and it forms archisporial cells or archisporium. So this archisporium, it undergo divisions. We know about the parietal divisions, and like that, it undergo the divisions and they form primary parietal cell and primary sporogenous cells. The same development like male gametophyte. Now, the primary parietal cells are also formed, the primary sporogenous cells are formed. Here, the primary sporogenous cells, which is inside, it will become the megaspore mother cell. The primary sporogenous cells, it becomes megaspore mother cells. This megaspore mother cells, it will undergo meiotic divisions and forms 4 cells. It forms 4 cells. The nucellus, one cell of the nucellus, it enlarges and become archisporial cells. These archisporial cells, it undergo divisions and forms primary parietal cell and inner primary sporogenous cells. These sporogenous cells are, it will become the megaspore mother cells. These megaspore mother cells, it undergo the divisions, meiotic division, and forms 4 haploid cells. In the case of male gametophyte, we have learned that the 4 haploid microspores, they become the pollen grains. I'll show you the picture. Same type of divisions. The microspore, it undergoes meiosis and forms 4 microspores. All the 4 microspore develops into pollen grains. Whereas in the case of gynoecium, there are 4 megaspores are formed. 4 megaspores are formed. Among that 4 megaspore, only one, it will become functional. This is the spore mother cells. It forms 4 haploid cells. Among that, 3 cells degenerate, and only one cell they develop into megaspore or embryo sac. This is called monosporic type of development in megaspore. Out of 4 haploid cells that are formed, 3 degenerate and 1 develops into megaspore or embryo sac. That's called monosporic. In bisporic, 2 haploid cells. 4 will be formed, 2 degenerate, 2 will develop into embryo sac. That is called bisporic. If all the 4 are involved in formation of megaspore, it is called tetrasporic. Children, are you listening to the class? Do you have any doubt till now? Are you clear with the developmental process? Any doubt? Vashini, Kadiravan, Balakrishnan, sure. Okay. So first என்ன ஆகுதுன்னு பாத்தீங்கன்னா ஒரு நியூசிலஸ் செல் ஆர்கிஸ்போரியல் செல்லா டெவலப் ஆகுது. இந்த ஆர்கிஸ்போரியல் செல் ரெண்டா டிவிஷன் ஆகி பிரைமரி பேரேட்டல் செல் ஃபார்ம் பண்ணுது, பிரைமரி ஸ்போரோஜினஸ் செல் ஃபார்ம் பண்ணுது. இந்த பிரைமரி ஸ்போரோஜினஸ் செல்ஸ் தான் மெகாஸ்போர் மதர் செல்லா மாறுது. இந்த மெகாஸ்போர் மதர் செல் மியோசிஸ் டிவிஷன் ஆகி 4 ஹாப்ளாய்ட் மெகாஸ்போரை ஃபார்ம் பண்ணுது. 4 செல் ஃபார்ம் பண்ணுது. இந்த 4ல 3 டீஜெனரேட் ஆயிட்டு ஒன்னு மட்டும் தான் என்னவா டெவலப் ஆகுதுன்னா only one will develop into the embryo sac. If one cell among the 4 haploid cells develop into megaspore or embryo sac, that is called monosporic. The non-functional spores die. It is degenerate. That's what means degenerate. It dies. From the 4 cells, if 2 cells are formed embryo sac or megaspore, that is called bisporic. If all the 4 together, it forms embryo sac or microspore, it is called tetrasporic. You can see the development here, how it is going to develop. The megaspore mother cells, it undergoes the meiosis process and forms 4 haploid cells. Among that, the 3 degenerate and 1 develop into functional megaspores. The functional megaspores, it undergo the process called mitosis cell divisions. And it becomes 2 cells. The nucleus divides. 2 nucleus. 2 become 4. 4 become 8. One functional megaspore, it undergo mitosis and they become 2 nucleus, divides into 2. And the 2 nucleus divides into 4. 4 divides into 8. See how they are arranged here. 2 it will be at the center, and 3 it will be here, and 3 it will be here. And these structures, it forms the embryo sac. This is actually the embryo sac. The 3 nucleus that is here, it becomes 3 cells. Those 3 cells are called as antipodals. 3 cells are called as antipodals. And here 3 cells are there, right? Among these 3 cells, 2 it will become synergids and 1 it will become egg cell. Whereas at the center you can see the polar nuclei. 2 cells it will be there. So altogether you can see it here, 1, 2, 3, 4, 5, 6, 7 cells with 8 nucleus. This is one cell, central cell, which contains 2 nucleus. Whereas these cells, synergids, it contains one cell. These are all 2 cells. X cell is 1 and antipodals 3 cells are there. So altogether We have 1, 2, 3, 4, 5, 6, 7 cells with 8 nuclei. This is a typical structure of the embryo sac. Typical structure of the embryo sac. So now we can see that how the embryo sacs are formed. This embryo sac, we call it as the megaspore. The megaspore. In that, we can see the egg cell, right? The egg cell contains the female gamete. The egg cell contains the female gamete. This is about development of monosporic embryo sac. Monosporic. We know what is monosporic, right? This is the monosporic embryo sac development. Okay, children. So this is the bisporic. You can see the bisporic here. 1, 2 cells. Now the 2 cells, they form the megaspore. This is 2. If all the 4 forms, it is called tetrasporic. See the development here. In monosporic, microspore mother cells undergo meiosis. 4 cells are there. The 4 divides into the functional megaspore, divides 2 nucleus, 4, 8, and 8s are arranged like this. Here, 2 cells, 2 become 4, 4 become 6 here. And they have arranged like this. And this is tetrasporic, where you can see that all the 4 functional all the 4 haploid cells, it will become functional megaspore. They develop like this. So now we can see that the flower, it has megaspore mother cells. That megaspore mother cells forms the embryo sac. The embryo sac contains the egg. Here, egg is here. This is the female gamete. Whereas the anther contains pollen grain. The pollen grains forms the male gamete. In next class, I'm going to explain to you about the pollination and pollinating agents. And after that, I'm going to explain to you about the fertilization process, fertilization in plants, how fertilization process takes place, how the embryo sac, it becomes embryo and endosperm, and development of endosperm, development of dicot embryo, and then seed. All these things we are going to learn in next class. Okay, children. Till now, if you have any doubt, now we can clarify your doubts. Yes, Madhav Krishnan, is endomitosis process because it occurs within the cells. Endomitosis. You're right. Any other doubt, children? Children, Saturday morning time, are you all free? Bisporic, here Balakrishna, in bisporic, they form, actually you can see it here, it won't form 7 cells and 8 nuclei. They form 4 cell with 6 nuclei. See it here, 1, 2, 3, 4, 5, 6. 4 cell with 6 nuclei. And tetrasporic, it forms n number of cells. We cannot count it, how many it is there. That is the difference. Even Saturday morning, are you all free? Yes, megaspore mother cells undergo meiosis and forms 4 haploid cells. Even microspore mother cells also undergo meiosis because any formation of gamete, it is meiosis process. Saturday morning, are you all free? Saturday morning, at what time you will be free, children? All of you. Saturday morning, at what time you will be free? I'm talking about Saturday morning. Vashni, Saturday காலையில எத்தனை மணிக்கு ஃப்ரீயா இருப்பீங்க? Children, just tell me at what time Saturday till what time you will be free? Say to any time, it's fine. Just you tell me what time you will be free. I'll plan according to that. Pasetu Laya. Deepak Ishwar, 9 to 2 school irukama. I have school from 9 to 2. Vashini, at what time you'll have math class? Okay, what I'll do is Saturday morning. Saturday morning, eight AM to 9:30 AM we'll have class. Will it be fine for everybody? 8 AM to 9:30. Saturday, 8 AM to 9:30. Will it be fine for everybody? Deepak, அந்த half an hour மட்டும் கொஞ்சம் adjust பண்ணிக்குமா? Okay, ma. So when I have a class on Saturday, 8 AM to 9:30. Okay, children, fixed. Thank you very much, children. Thank you.